What if I told you that staring at a blank wall for just 3 minutes could literally rewire your brain for the better?
I mean, it sounds completely counterintuitive, right?
It really does. Or, what if I said that the secret to controlling your mood, avoiding brain fog, and protecting your cognitive health is actually pulverized dead bacteria living inside you?
Yeah, that one definitely sounds like science fiction.
Right. But welcome to this deep dive. We are taking a massive stack of research today, papers, clinical notes, and this really fascinating overarching framework called Foundations of Cognitive and Metabolic Longevity. And, well, we're extracting the gold.
Exactly, because our mission today isn't to give you, just another exhausting list of random health tips.
No, we know you want to cut through the noise.
Yeah.
Today is really about uncovering the surprising interconnected mechanics of how your brain and body actually function behind the scenes.
Right, because the research we're looking at, it completely challenges the conventional wisdom of what it means to be well-informed about your own biology. We're stepping away from superficial symptoms and looking straight at the why.
The underlying gears of the machine. Okay, let's
Yeah.
unpack this because the blueprint of what we're covering today is wild. We're going to explore everything from the neurological necessity of pure agonizing boredom to how, common grocery store lemons can short-circuit the brain's addiction pathways.
It's a journey, for sure.
Yeah, so let's start right at the top with the physical brain and this concept called the 3-minute hold.
Oh, this is fascinating.
The research suggests that when you're just, say, standing in line at the grocery store and you feel that uncomfortable, almost magnetic itch to pull out your phone, you have to resist it. You're supposed to just stand there and be bored for 3 minutes.
Yeah, because there is a hard, measurable neurological mechanism at play here. When you stop consuming external information, a specific network in your brain finally gets to power up. It's called the default mode network, or the DMN. And this is the system responsible for your deepest creativity, your empathy, your ability to simulate future scenarios, and your self-reflection.
But it's an incredibly shy system.
Right, super shy. It only comes online when external consumption completely stops. Boredom is the absolute prerequisite.
It makes me think of the background defragmentation software on an old computer.
Oh, that's a perfect analogy.
Right, like it can only run and organize all your fragmented, messy files when you physically step away from the keyboard. The moment you move the mouse, the defrag pauses. So are we essentially pausing our brain's defrag process every time we look at a screen?
We absolutely are. I mean, cognitive researchers point out that if you are constantly shoving external stimuli like short-form videos or emails into your visual cortex, the DMN stays completely dormant.
So the brain never gets to file its folders away.
Exactly. And we see the power of this in this really interesting university study. Researchers forced participants to do an agonizingly mundane task. They had them literally copy numbers out of a phone book by hand.
Oh, wow. That sounds terrible.
Right. But following that period of intense boredom, those specific participants dramatically outperformed the control groups on creative thinking tests.
Because their brains finally had the empty space required to connect disparate dots.
Exactly.
But that raises a crazy question, though. If taking a few minutes to let the DMN run is so beneficial for our creativity and our resilience, why is it so incredibly hard to do? Are we really that terrified of our own thoughts?
Well, the data suggests we absolutely are. There's this famous university study that set out to test this exact discomfort. They placed participants in a completely empty room for 15 minutes.
Just an empty room?
Yeah, no phones, no books, no windows. The only object in the room was a button. And if you pressed the button, it delivered a painful electric shock.
Wait, they were told beforehand that the button would physically hurt them, right?
They were. They even experienced a test shock first, so they knew exactly how much it would hurt.
That's wild.
And despite knowing the pain, 67% of the men and 25% of the women actively chose to shock themselves rather than just sit quietly with their own thoughts.
You're kidding.
Nope. One guy actually shocked himself 190 times in 15 minutes.
He chose physical pain over mental stillness 190 times.
Yeah, but that chronic interruption, that constant flight from boredom, it actively harms long-term resilience. It keeps the brain in a state of chronic stress.
Right, because it prevents the deep self-reflection required to actually process emotions.
Yeah.
I can completely see how that mental rest protects the brain's function.
Yeah.
But I want to pivot here because the research stack transitions into something pretty alarming.
The physical structure of the brain.
Yeah. All that mental rest doesn't mean anything if the actual hardware of the brain is literally corroding.
Right.
Let's talk about homocysteine. The sources say everyone should be testing this blood marker, aiming to get it below 8 micromoles per liter. But I have to push back here. We've been told for half a century that cholesterol is the ultimate villain for our arteries. Are you saying the medical establishment has been focusing on the wrong target this whole time?
What's fascinating here is the forgotten history. Back in 1969, there was a Harvard researcher who was studying children with a rare genetic disorder. And these children were dying of massive strokes and advanced atherosclerosis before they even reached the age of 10.
Before age 10. That's awful.
It is. But when he examined their blood, it wasn't cholesterol causing the massive plaques. It was an astronomical buildup of homocysteine.
So it's not just a passive marker, it's an incredibly potent toxin.
It is a direct active toxin. Medical pioneers have mapped out exactly how it works. Elevated homocysteine drives massive oxidative stress in the blood.
Think of it like microscopic rust, right?
Exactly. Rust that actively destroys the delicate inner linings of your arteries. And more concerning for longevity, it drives a metabolic reprogramming that specifically targets and damages vulnerable gray matter in the brain.
The statistics in the notes are sobering. Meta-analyses show that for every 5-point increase in your homocysteine levels, your risk of Alzheimer's disease goes up by 12%.
And your risk of a stroke skyrockets by 60%.
It's basically rusting your brain and your blood vessels from the inside out. But the research points out that if you actually test for it and catch it, the fix is shockingly simple.
It really is. The fix is just highly specific, active B vitamins. We are talking about methylfolate B12 in the form of methylcobalamin and B6.
And these vitamins act as the chemical machinery that recycles that toxic homocysteine back into safe amino acids.
Right. The most compelling evidence comes from a major clinical trial where they took patients and simply gave them these B vitamins. They found it slowed whole brain atrophy, literal brain shrinkage, by 30% compared to a placebo. And for the patients who started the trial with highly elevated homocysteine above 13, the brain shrinkage was slowed by a massive 53%.
That is profound. You are cutting the physical aging of your brain in half just by clearing out a rest-causing toxin with a few vitamins.
Exactly.
But homocysteine is a long-term toxin. The sources point out there is another everyday component in our blood that damages neurons in real time when it spikes too high, and that's glucose, sugar.
This is a critical metabolic mechanism because neurons absolutely prefer glucose, right? They need it for fuel. But the damage occurs during massive, unnatural spikes.
Like when you eat a huge plate of pasta.
Yeah, or a sugary pastry. It creates a sharp vertical spike in your blood sugar. And this intense rush triggers specialized sensors in your gut called neuropod cells.
Okay, let me guess. These neuropod cells have a direct hotline to the brain's reward center.
They do. They flood the brain's mesolimbic pathway with dopamine. To put that in simpler terms, they hit the brain's addiction superhighway.
Which evolutionarily was a brilliant survival reward for finding dense calories in the wilderness.
Right. But in the modern world, this dopamine flood just drives uncontrollable cravings and leads to the long-term metabolic dysfunction that fuels obesity and neurodegeneration.
Well, I saw in the notes that Andrew Huberman was doing some crazy personal experiments with his continuous glucose monitor to see if he could hack this exact spike.
Yeah, he was running a fascinating self-experiment. He wore a CGM and intentionally ate large, carbohydrate-heavy meals to induce those destructive blood glucose spikes.
Sounds dangerous.
Well, he introduced one variable. He would ingest a couple of tablespoons of lemon or lime juice in water right around the meal. And his real-time CGM data showed that the acid from the citrus fundamentally blunted his blood sugar response.
So the massive spike flattened out into a gentle rolling hill.
Exactly.
Here's where it gets really interesting, because the mechanism of action is actually a two-part system. First, there's a neural trick. The sour taste receptors on your tongue act like a biological circuit breaker. When that intense sour flavor hits, it physically alters the neural response to the sweet food you're eating, basically short-circuiting that massive dopamine release. You simply don't get that immediate addictive high.
And then the second part happens post-ingestively, down in the stomach. The acidity of the lemon juice physically slows down gastric emptying.
So it acts as a biological speed bump.
Exactly. Instead of dumping all the digested sugar into your intestines at once, it releases it slowly, controlling the rate at which the glucose enters your bloodstream.
So you still get the energy from the food, but you spread it out over time instead of overwhelming the system.
Yeah.
But, this whole discussion makes me wonder. You wouldn't need a citrus speed bump for sugar. if you didn't have intense, uncontrollable cravings for junk in the first place.
Very true.
I'm looking at these clinical notes here about women heavily restricting to 800 calories a day to lose weight. I don't get it. If they have the sheer willpower to starve all day, why are they suddenly binge-eating cookies in the pantry at 9 p.m.?
Because their bodies are literally starving for structural building blocks, and the brain basically takes over to keep them alive. This is known as the protein leverage hypothesis.
Okay, what does that mean?
The foundational principle is that your body will actively seek out food and subconsciously drive you to keep eating until its foundational protein requirement is met.
Oh, wow.
Yeah. If you are feeding it 800 calories of salads and low-fat crackers with almost no protein, your body leaves the hunger switch permanently flipped on, hoping you'll eventually stumble across a steak or some eggs.
So the body doesn't actually care about the calorie count.
Yeah.
If it doesn't get the building blocks, it forces you to eat.
It forces you to overeat processed fats and carbohydrates, trying to satisfy that biological drive. Nutrition researchers highlight a specific target here. You need roughly 100 grams of high-quality protein a day.
And notice say that once these patients simply prioritized hitting that 100 grams early in the day, the 9 p.m. cravings completely vanished.
Because the body was finally structurally nourished, so it turned the hunger switch off.
That makes so much sense. And the research adds a critical component to this protocol. They found that higher protein diets independently preserve your metabolic rate and yield a much stronger, longer-lasting satiety signal than fiber alone.
Though the sources are clear that you must backfill that protein with 25 to 35 grams of whole food fiber.
Right, because the fiber physically slows digestion and naturally lowers your daily caloric intake by about 10%.
And beyond just feeling full and preventing binges, hitting that protein goal is what preserves and builds your muscle mass. Muscle is critical. It prevents age-related sarcopenia, which is just the natural wasting away of muscle as we age.
But more importantly for your longevity,
Yeah.
muscle acts as a massive metabolic glucose sink.
Yes, meaning it's the physical location where all that blood sugar actually gets burned up safely.
Precisely the point. Maintaining that muscle keeps your insulin sensitivity high and stops your body from panicking and storing excess sugar as dangerous visceral fat around your internal organs.
Okay, so muscle acts as a metabolic sink for our daily glucose. But what acts as the sink for the physical toxic junk we are exposed to?
Yeah, that's a huge issue.
I'm talking about the microplastics in our water and the environmental toxins we inhale every single day. The research points to something called stabilized sulforaphane, extracted from broccoli seeds and sprouts.
Now, I have to ask, broccoli sprouts. Is this just another internet detox fad, or is there real cellular mechanics going on here?
It absolutely sounds like a fad until you look at the hard science. Researchers explain that sulforaphane is the most potent natural activator of something called the NRF2 pathway.
Okay, what is NRF2?
Think of it as a master switch inside your cells. When sulforaphane flips that switch, it instantly turns on over 200 different antioxidant genes and drastically upregulates your liver's detoxification process.
Wow.
But the most incredible part of this mechanism is what it does to the lysosomes.
Oh, I remember from high school biology that lysosomes are basically the cellular garbage disposals. Right.
That's a great way to picture them. Sulforaphane actually triggers these cellular garbage disposals to gather up trapped toxins and embedded microplastics.
Wait, it can flush out microplastics?
Yeah. It forces the lysosomes to physically fuse with the outer surface of the cell and mechanically flush that garbage out into the lymphatic system so your body can excrete it.
The data on this was wild. Just 10 milligrams of stabilized sulforaphane decreases a major inflammatory marker called IL-6 by 30% in just 24 hours.
Which is huge because IL-6 is essentially the body's fire alarm. If IL-6 is high, your immune system is panicking. Lowering it means putting out the fire.
But the notes specify it has to be stabilized sulforaphane, not a precursor like glucoraphanin. Why doesn't the precursor work?
Because glucoraphanin is just the raw, inert material. It requires a highly specific enzyme to convert into active sulforaphane in your gut. And if your gut microbiome isn't perfectly optimized, which most people's aren't, the raw material passes right through you and does absolutely nothing.
I see.
Stabilized sulforaphane is the active, finished product ready to work immediately. It even physically seals the tight junctions in your gut, patching the microscopic holes so toxins can't leak back into your blood.
There's an astonishing real-world story in the notes about a lysosome researcher who decided to test a massive dose of stabilized sulforaphane on himself.
Oh yeah, he decided to be his own guinea pig.
What happened?
Well, he took the dose, and the very next day, his lab tests showed the absolute largest release of microplastics into his blood and urine that the lab had ever recorded in any human subject.
His body was violently dumping the plastic.
Exactly. And by day 2, his microplastic levels dropped below his original pre-experiment baseline because his system had successfully flushed it all out.
That is unbelievable, just flushing the plastic right out of the cells. But, clearing out the microplastic is really just getting rid of the drag on the system. It frees up the cell, but how do we restart the engine? How do we get our energy production back to youthful levels?
This is where we look at the mitochondria, the tiny power plants inside the cells.
Uh,
As we age, we experience a phenomenon called inflammaging.
Inflammaging.
Yeah, it's a low-grade chronic swelling of our microcirculation, our tiniest capillary blood vessels.
Wait, so even if I'm breathing heavily and my lungs are full of air, the oxygen isn't actually reaching the cells?
The vessels are literally swollen shut. The red blood cells physically cannot squeeze through to deliver the oxygen to your tissues.
That's terrifying.
It creates a state of pseudo-hypoxia. Your tissues are suffocating, forcing your cells into a highly inefficient anaerobic state where they can't produce enough ATP, which is pure cellular energy.
And as the experts point out, oxygen is the true limiting currency of biological aging.
Right, so the protocol here to force the oxygen back in is a 1-2 punch. First, 15 minutes of something called EW exercise with oxygen therapy.
So you do intense cardio while breathing highly concentrated pure oxygen from a massive reservoir bag.
Exactly. The heavy exercise forces your blood vessels to dilate wide open, while the supplemental oxygen completely saturates your blood, driving it deep into those previously swollen, suffocating tissues.
And then comes the critical second part. Immediately after the EW session, while your heart rate is still elevated and your blood is saturated, you do a full-body red light therapy session.
Right, because the red and infrared light photons actually penetrate the skin and act like jumper cables for the mitochondria.
The light physically pushes extra electrons directly into the cells. It turbocharges the mitochondria so they can rapidly produce that ATP energy.
And because you just flooded the whole system with oxygen during the workout, the mitochondria don't run out of fuel while they're working in overdrive.
It's a profound, synergistic combination. Dave Asprey tells a compelling story about the power of this mechanism.
Oh, the muscle knot story.
Yeah. He suffered from a painfully tight, completely hypoxic muscle knot between his shoulder blades, triggered by toxic mold exposure. He tried every kind of deep tissue massage, but nothing worked because the tissue itself was physically starved of oxygen. It was deadlocked.
Right, so what did he do?
He finally used a powerful infrared laser directly on it. The infrared light penetrated the tissue, restored the capillary blood flow, pushed oxygen back into the star cells, and instantly melted the knot away.
That's incredible.
It even warmed up his hands, which had been chronically cold for years due to poor microcirculation.
Okay, let's step back and look at this entire massive physiological network we've discussed. We've talked about optimizing the cells with oxygen, flushing out the microplastic junk, preserving muscle as a glucose sink, and protecting the brain from homocysteine rust.
But who is actually the CEO running this entire operation? Who is pulling the levers?
Well, if we connect this to the bigger picture, the CEO isn't actually located in your head, it's in your gut. Your microbiome acts as a literal second brain.
And it's directly wired to your primary brain via the vagus nerve, right?
Exactly. The vagus nerve functions like a massive, high-speed fiber optic cable, constantly texting chemical updates back and forth.
And the research says we need to be feeding this gut system with fermented foods and polyphenol-rich plants. But so what does this all mean when the sources explicitly say we should be feeding the graveyard microbiome?
The graveyard microbiome.
Yeah, that we shouldn't worry if the probiotic bacteria in our fermented foods are already dead. How can dead biological matter do anything in the body?
It sounds completely counterintuitive, but it's one of the most exciting recent discoveries in longevity research.
Yeah.
Science used to assume probiotics had to be alive to colonize your intestines and provide any benefit.
Right.
But it turns out the carcasses and the rigid cell walls of dead bacteria are highly biologically active. They still possess the exact structural signaling molecules required to send messages up that vagus nerve to your brain.
So think of it like a physical key fitting into a lock. Even if the hand holding the key is gone, the shape of the key still turns the tumblers.
That is exactly it.
There's an MIT study that perfectly illustrates this, and it really blew my mind. Researchers were studying a specific strain of bacteria called Lactobacillus reuteri. They knew that when they fed it to animals, it triggered the brain to release oxytocin, the social bonding hormone, and it literally doubled the rate of physical wound healing.
Right. And to test the graveyard theory, the researchers did something drastic. They completely pulverized the live bacteria using destructive nasal ultrasound
They absolutely obliterated it.
to ensure every single bacterium was completely dead.
Wait, pause. They completely obliterated the bacteria. It was just pulverized, dead biological material, and when they fed it to the animals, it still released oxytocin in the brain. How is that even physically possible?
Because the physical fragments of the cell walls survived the ultrasound. Those structural molecules locked into the gut receptors and successfully pulled the chemical levers in the brain. Which is why optimizing your gut environment is so crucial to your mood. There's a story in the clinical notes about a 9-year-old patient who suffered from severe violent behavioral issues. She was constantly getting kicked out of school for erratic behavior.
Oh, that's sad.
The doctor ran a comprehensive stool test and found a massive fungal and bacterial overgrowth in her gut.
So her dysfunctional gut was sending constant panic signals up the vagus nerve to her brain.
Exactly. He simply cleared out the bad bacteria to restore the microbiome balance, and she transformed overnight into a calm, sweet child.
Unbelievable.
It proved to him that irritable bowel causes irritable brain, not the other way around. Optimizing the CEO system reduces neuroinflammation, which is really the root cause of both behavioral issues and long-term cognitive decline.
So, to synthesize this incredible journey we've just been on, it starts with the absolute necessity of sitting bored in a line to let your default mode network defragment your mind. It's aggressively checking your homocysteine to physically protect your gray matter from rusting. It's using a shot of lemon juice and hitting 100 grams of protein to hack your dopamine cravings and build a metabolic glucose sink.
Yep.
It's flushing out microscopic plastic with stabilized broccoli sprouts, forcing oxygen into suffocating mitochondria, and realizing that the pulverized dead bacteria inside your gut are secretly the ones pulling the strings of your mood and your long-term health.
It really completely reframes how we view our daily habits, and it leaves us with a profound final thought to ponder.
Oh, I'm ready for it.
What if our modern environment, the constant smartphone stimulation that kills the DMN, the hyperpalatable processed foods that spike our glucose, the invisible microplastics in our water, and the indoor sedentary lifestyles that starve our tissues of oxygen, what if this modern world is perfectly designed to disrupt every single one of these natural longevity mechanisms?
That is a scary thought.
Are modern biohacks just a desperate attempt to artificially simulate the natural world our bodies originally evolved to thrive in?
We really do have to work incredibly hard just to give our biology what it naturally expects. We encourage you to take just one insight from this deep dive, whether it's the 3-minute hold the next time you're in a waiting room, or the lemon juice trick before a heavy meal, and experiment with it today. Thank you for learning alongside us.